Okay, good afternoon, everyone. So it's a pleasure to be here and have this session devoted to the understanding of HTT function. And I will call the first speaker Stefan Kochanek, who's a professor at the Department of Gene Therapy at Ulm University. So Stefan, the stage is yours.
Yeah, thank you very much. So. Okay, so let me see how that works. So I've been asked to talk about the pathophysiology of HD and whether Huntington HAP40 complex might play a role there. We heard this morning very interesting data and concepts about different disease mechanisms and how they intersect and work together to lead to HD. So, as I said, today we will have a look at the two proteins. And it took 25 years, actually, from identification of the gene causing HD to have actually a high-resolution structure of the protein. So it started basically by a bioinformatical analysis in 1995 when certain structural elements, which are called heat repeats, antiparallel alpha helices, were identified. Then in 2006, that was the first time when full-length Huntington was produced in insect cells, and one could see particles by a transmission electron microscopy, and also by TEM, then the first low-resolution structure in 2016 was obtained at 32 angstrom resolution. And so in 2018, then the structure of full-length Huntington together with HAP40, Huntington-associated protein 40, and then even at 4 angstrom resolution, and in the last year, even higher resolution at 2.6 by Rachel Harding and colleagues. So the HAP40 is a very abundant interactor of Huntington, and I would say, so this gel has been a very fortunate gel. When we try to produce in cell lines, stable cell lines, full-length Huntington to maybe do structural studies, when we saw that when we isolated the complex, we saw this band, which turned out to be HAP40. And when purifying the process, the complex, by size exclusion chromatography, one could see that this protein in blue eluted like a well-behaved soluble protein, different from full-length Huntington itself in red, which forms aggregates quite easily in tubes. And also by differential scanning fluorimetry, having here a very nice melting point with the complex, but not with Huntington, it was clear that this complex might be amenable to cryo-electron microscopy. And so basically, before it was not possible because the Huntington by itself was just too heterogeneous, too flexible. But when in combination with HAP40, we got this quite rigid structure so that cryo-EM actually was possible. And for those, and this has been then the first data on the high-resolution structure, and for those who are not so familiar, haven't maybe seen the data. So the Huntington is basically divided in 3 domains. The blue one, which is N-terminally located, which we call N-heat, a C-terminal located domain, which we call C-heat, and then we have a yellow bridge domain, which connects the two. And in between, there's the HAP40 connecting with all 3 domains. And let's have a look, which is also important for the discussion in later slides. So how the interaction of HAP40 with Huntington is. So there's basically two types of interaction. Over a very large extended area interacts the HAP40, that's like an open book view, mainly by hydrophobic interactions with the Huntington, and with these two domains. And then in addition, we have an interaction of the HAP40 with the bridge domain by electrostatic interactions. So we have negative amino acids here on the HAP40 side and positive amino acids here on the Huntington side. The question is HAP40 essential? And does HAP40 play a role in HD? So what had been known so far, that it's a very abundant interactor of Huntington, and actually this has been seen in vivo in mouse models where HAP40 has been the most abundant interactor of Huntington. The structure which we determined showed a TPR type of structure. That's proteins which are frequently HAP proteins, or they interact with many different proteins. So that is this. And there has been one publication that HAP40 might mediate the interaction of Huntington with a GDPase RAP5, which is involved in vesicle transport. But they have very few publications actually on this very abundant interactor of Huntington in the past. So today I would like to discuss the interaction of HAP40 with polyQ expanded Huntington, have some words on the evolution of HAP40, on expression and stability, and some questions and future research areas. So we analyzed a full-length Huntington with 17q, this I showed you before, but also with 46q and 128q, and also a protein in which the polyglutamine had been deleted. And by all biophysical methods, size exclusion, chromatography, differential scanning, fluorometry, or cross-linking mass spectrometry, there was no difference between any of those. And also by cryo-EM, there has been, in our hands, we have not seen any difference. Rachel Harding also published very similar data, also with some modeling on the disordered parts of the protein, and maybe suggesting that under certain conditions there might be some interaction with expanded polyQ with some disordered protein regions. But I think it's not really clear. But overall, the structure of Huntington in a complex with HAP40, I would say, is pretty much identical. What about evolution? Evolution, also as we heard this morning, might give some sense on whether this protein might in principle be essential. So we looked at the evolution of the factor VIII gene, locus, that's the gene which codes for the protein HAP40. And there's a few interesting things. So first of all, in non-amniotes, the factor VIII gene is a multi-exon gene between at least 2 exons, or very frequently up to 15 exons. And it's autosomally located. While in amniotes, it's X-chromosomally located, and it's a single exon gene. So from amniotes, non-amniotes to amniotes, a conversion took place to a single exon gene. And also very interestingly, then the single exon gene amplified in some species, so that in humans we have 3 copies, and also orangutan, and in some of mammals we have the others, we have between 1 and 2 copies. In all other species, there's only 1 paralog. So the mechanism is that the conversion likely took place by retroposition, so integration of a reverse transcript. And it's quite interesting that normally neutral duplications which do not have an effect do not get fixed normally. But here what happened is the multi-exon gene during the conversion got lost actually, and the single exon gene survived, and even then amplified in some of the higher species. So I think that's very interesting and might indicate that this might indicate maybe some increased evolutionary fitness by the copy number increase. And the other thing is that HAP40 strictly co-occurs with Huntington to the roots of unicorns with very, very few exceptions. That's also very interesting. Also, the core sequences of Huntington and HAP40 have higher conservation scores than the intrinsically disordered parts of the proteins, which is actually not really surprising. But I think it was very interesting that the charge complementarity of this charge clamp between HAP40 and the bridge domain of Huntington is highly conserved. So this suggests that this interaction actually takes place also in lower species, and the interaction of HAP40 and Huntington we have biochemically confirmed this in zebrafish, and it has also been shown in Drosophila by Shen Zhang's group, very recently published. And also there is some functional data from this publication that the HAP40 knockout in Drosophila has a similar phenotype like Huntington. It's like mainly an aging phenotype, and also that HAP40 can modulate the toxicity of Huntington disease models in Drosophila. There's one also interesting homology, actually, that the HAP40 and the NSF attachment protein family, there's 3 in humans, 1 in yeast, is very high. The homology is very high. So they are both by bioinformatic analysis, evolutionary and also structurally related. So these proteins are cytosolic adapter proteins which are important for vesicle fusion at membranes. So what about the stability and expression of HAP40? So this also was like an eye-opener experiment when we made cell lines, stable cell lines, which allowed inducible expression of Huntington. So if we induce expression of Huntington, the levels of HAP40 strongly increase. So that is without doxycycline, that is with doxycycline. And also we could see by some translational blocking experiment that this interaction with Huntington obviously is post-translational, and we could see that the half-life of HAP40 in the presence of Huntington is strongly increased. So as a summary, we have endogenous and also transgenic HAP40 levels increase upon Huntington induction. The increase of HAP40 levels following Huntington induction is actually independent of poly-Q lengths. I didn't show you the data, so that is independent of poly-Q lengths. And the stability of half-life of HAP40 is increased upon Huntington induction due to binding physical interaction with Huntington. What about HD? So we looked at HAP40 and also, of course, Huntington levels in different cell types. So we looked in human fibroblasts of HD patients and of controls. Now, it has been known that Huntington levels, there's several publications, Huntington levels are reduced in the course of the disease and depending on the length of the poly-Q. But what we saw here is that also HAP40 levels actually are decreased. And that is quantified here, as you can see here. So HAP40 levels and Huntington levels both correlate. They are both significantly increased. When we do the same experiment in human lymphoblasts from Huntington disease patients, we see basically the same thing. So both Huntington levels are decreased and HAP40 levels are decreased. And also, based on published data by the lab from William Young, where we looked a little more closely, we saw that when looking at the mouse proteome of the striatum and also of the cortex, so Huntington levels are decreased, but also HAP40 levels are decreased. So what we know is now, as a functional consequences of Huntington HAP40, we have a conserved interaction and a co-evolution likely between HAP40 and Huntington. The HAP40 levels depend on Huntington protein levels, and I cannot really see this. Yes, and the HAP40 half-life is increased together with increased Huntington levels. In patients, we see that both Huntington levels and HAP40 levels are decreased. And there's maybe some indication on this one publication, 2006, interaction maybe with RAP5, and also on the structural and evolutionary similarity, a potential role in vesicle transport. One thing I didn't mention, actually, there's one additional interesting thing which we are looking at what that might mean actually. In mammals, actually, there's in the center of HAP40 a polyproline tract inserted, only in mammals. In non-mammals, it's not present. So now what could be the function? And let's speculate a little bit of HAP40 in health and disease. Now, one thing could be that now the complex might be sort of an inactive storage form of Huntington. So we could think that maybe HAP40 might play a role in simply keeping Huntington soluble in the cytoplasm. That has me maybe my initial thought. I'm not so sure about this now, because I think probably HAP40 has some function, other function outside of that. But it might actually influence the subcellular localization of Huntington and also might influence Huntington function. And also there might be a specific function of the complex versus the individual proteins. On the other hand, the complex might be an inactive form of HAP40, obviously, and the subcellular localization of HAP40 might be influenced by that interaction, and the HAP40 function might also be affected. So it will be very interesting also then to see now, to look more specifically. So far the data on protein-protein interactions have mainly been with Huntington and hundreds and maybe thousands of potential interactors, to see what is actually interaction between these other proteins and Huntington, and what is the one actually which interacts with the complex. So the question is really, what are the functions? If it would be sort of just a passive protein, sort of shielding the hydrophobic surface of Huntington to keep it soluble in the cytoplasm, then maybe one would not need to think more about the function of HAP40. But as I said, and we have also some data from overexpression studies, etc. So I think there is probably much more behind actually HAP40. So now I start to believe it probably has some other functions as well. So it will be very interesting to see what are the proteins binding to the complex rather than to Huntington alone, and what is the situation in case of polyQ expansion. And very importantly is the decrease of HAP40 protein levels in HD pathogenically relevant. So that's, I think, is a very interesting questions. We know Huntington levels can be lower, 50%, no problem. But what about HAP40? And one suggestion from Bernhard Landwehrmeyer, whether HAP40 could be maybe used as a biomarker. So that's for my part, and these are my thanks to the people who did the work, Manuel Seefelder, Bin Huang. Then we're working with Fabrice Klein and Katrin Lindenberg in Bernhard Landwehrmeyer's group, evolutionary studies together with the MPI in Tübingen, André Lupasch, and cryo-EM studies actually were done at the Max Planck Institute in Munich, and further collaborations. So thank you very much.
Thanks, Stefan. Very interesting talk. Question from the audience? Yeah, there is a question there.
Stefan, very nice talk. I'm here. Stefan? Hi, it's Eric. Very nice talk. Stefan, I have a question. Do you have any evidence? I agree with you very, very much in terms of the free Huntington or complex or free Huntington complex and complex function, but maybe also with free HAP40. So do you have any evidence that under endogenous conditions in a cell model or other or neurons that there is free HAP40 flying around and maybe doing something independent of the Huntington protein, or what are your thoughts about that?
We need to look at this very carefully. Of course, if we overexpress HAP40, you will see a lot of things. So a lot of the HAP40 then goes into the nucleus, for example. But we tend to believe that there will be HAP40 by itself, so where it does not co-localize with Huntington. But part of the problem has been that there has not been very good antibodies, for example, for the detection of HAP40. That's why we have been generating some, and hopefully very soon have some good monoclonals to follow the protein. So no clear answer. My guess is probably yes. Yeah.
There is another question over there. Yeah.
Thanks, Tom Massey, Cardiff. I was just wondering whether you know if HAP40 interacts with any of the known genetic modifiers of Huntington's.
We don't know yet. But we are in the process of doing interaction studies at a very systematic level. So probably in a few months, I could answer the question better than now. Yeah.
Thank you, Stefan. And now it's time